This time you're in your living room.
Hmm.
Last time you're in your own room, like with your whole setup.
Oh, it's like the same room. All right. All right. So today, Charlotte, we've been talking
Brother,
about.
what lesson is this?
Yeah, this is 3.1.
Okay.
All right. So we're talking about the cell cycle. So we're gonna talk about like how cells grow and then how they divide, right? So some of you guys have heard about things like mitosis, meiosis, stuff like that, right? So so we're gonna talk about like how cells grow and then how they divide, right? So you can kind of see here like this circle, it kind of so it's called a cell cycle, right? Because it goes around and around and around, right? So once a cell divides, then how does it go through the process, right? It goes through the circle again, right? So this is called a cell cycle because all the cycles repeat. Um, when a cell divides, then the new cells they go through the cycle again, so the whole process is repeating, right? So there's different kinds of portions. You can take a look at some of these. So we're going to talk about these in detail, but you can take a look. There's like different, you know, there's like different parts of the cell cycle, right? You have G1, then it goes to S phase, then G2, then it goes through mitosis and cytokinesis. That's where two cells actually form, where one cell actually turns into two cells. And then once you have those two daughter cells, then those cells, repeat through the cycle again and again, right? You have this thing over here called G0, right? Notice all here at the bottom you have G0, right? So that basically stands for, like, let's suppose, so most cells in the body are here in G0. Most cells in the body are not like constantly going through this cell cycle. They're kind of, this is where, the cells are kind of resting in a certain sense. They're not like growing and getting ready to turn into two cells. They're just sitting there doing their normal function, whatever it is that they were supposed to be doing, right? So most cells are in G0, but then from G0 they can go back into G1, right? And then, for example, like within mitosis, you'll notice over here, within mitosis, there's like a whole
Where
bunch of
does the
different.
circle start, brother?
So there's no starting, right? This is a circle. So circles don't start somewhere, right? So it just goes
Like,
around.
what's the first phase?
Hmm? It's not
Dude,
really
we'll
like a first.
find out.
All right, but it's not really like a first phase, right? It's like asking where does a circle begin, right? So it doesn't really begin anywhere, right? Right. So, all right. So this is just to kind of like give you like a couple of pictures about how um, right. So this again shows you that when the cells go throughout this cycle, right? So for example, let's suppose they go through from G1 to S to G2 to M, right? At certain points, there's like certain checkpoints, right? So when it goes to the checkpoint, the cell gets to decide, hey, am I ready to go to the next phase, right? Am I ready to keep going? So if that happens, then it can go on. Otherwise, it stops there and it doesn't go on, or it tries to get ready to go on, something like that, right? So so there's certain checkpoints throughout the cell cycle, right? Now it says here G0 again. Most of the cells are in G0, all the way here in the bottom left, right? So most of the cells are not like dividing and replicating their DNA and all this kind of stuff. Most are just sitting here in G0 and not doing a whole lot, right? So this is a very controlled kind of cycle, right? Like if the cell's not ready, it cannot go on to the next phase. Now does anyone know what is it called when when those checkpoints are not working? Like let's suppose the checkpoints are supposed to stop you from going to the next phase, right? But let's suppose you know you know everything has not been checked. There's still problems, but the cell just keeps going and dividing and forming more and more and more cells, even though you're not supposed to, even though it's supposed to be controlled. Anyone know what that's called?
No.
Any ideas? So that would be called like a cancerous kind of growth, right? So in cancer, right? What is cancer? Anyone heard of cancer? Like honest, what is cancer?
Isn't cancer like a disease?
It's like a disease, right? Yusuf, do you know like what happens in cancer?
Uh, I think depending on like wherever you get it,
Mm-hmm.
your cells like stop functioning or like they
Mm-hmm.
start functioning in weird ways.
Mm-hmm.
And
Yeah.
what do you call d cancer cells keep like duplicating and multiplying throughout your body?
Freeha, what do you think? What is cancer? What is a cancerous cell? Doesn't work properly, right? So I think Anas had a good idea. So it's like when a cell just keeps dividing and dividing and dividing, right? So they have like cancerous growth, right? And then the cancer cells they spread everywhere and start taking over everywhere, right? So normally, right, cells like skin cells, liver cells, any kind of cell, they grow when the body needs them to grow, and then as soon as it's done, it stops, right? Like let's suppose you cut yourself, right? The skin grows back, right? The skin cells they grow back, and then when that the cut is healed, basically they stop growing, right? You know, mm-hmm. Yeah. So that would be called cancer. Like if it just keeps growing, right? Even though it's not supposed to, even though the problem is fixed now, it keeps. Yeah. So you'd have like a big mass there, right? Like instead of the skin being nice and smooth there, all of a sudden that's going to be growing and growing and growing, and you have like a big chunk, like a big mass of skin there. So that's called like a tumor, right? So that's kind of what cancer is. So cells they should be going through this in a very controlled kind of process. They don't go on until they're ready. But if that doesn't happen, then that's called cancer, right? So we're going to start going through like some of the specifics, right? So the biggest, so you see this in the yellow, you see in the circle, they divide it into two parts. One is kind of like a yellowish orange kind of circle, right? And then one is the green circle, right? So the yellowish orange part, that's called interphase. That's like you see how that's the longest part of the cell cycle. Cell division, where the cells actually divide, that's a very short phase, right? So this whole yellow orange, arrow, this is called interphase. So that includes G1, S, and G2, right? All of this together is called interphase. G1, S, and G2. So this is the longest portion of the cell cycle, right? So then at the end of G2, at the end of interphase, then the cell can actually physically divide, right?
So then G1, S phase, and G2, these are all a part of the interphase, and then
Right. Mm-hmm.
mitosis, cytokinesis, and all of those are separate, like the
Correct.
actual cell division.
Right. Correct. Right.
So this is cell division, not cell reproduction, right? Because it's dividing, or you could call it. Or you could
Right.
also call it
Yeah.
cell
Yeah.
division.
Yeah. So you could call it the same thing. Yeah.
All right.
Right. So yeah, this is just another picture that's showing right interphase. Right. So interphase starts at G1 through S and G2. Right. And so then the mitotic phase is separate. Right. That's where the cell actually divides physically into two portions. Right. So most of the time the cell is an interphase. Right. So for example, it's giving an example here. Right. So it says, for example, a human skin cell will spend about 20 hours in interphase. Interphase can be divided into three phases. So usually interphase takes a long period of time, and then cell division occurs, much more quickly. Right? So interphase is the longest time of a cell's life. Right? So now let's take a look within interphase. Right? So within interphase, you have something called G1. Right? So G1, that's the first part of interphase. So G1, that's basically for cell growth. Right? Now you look over here on the right, right? So from G1, G0 is attached to G1, right? So from G1, a cell could go into G0 and just stay at rest and not be involved in cell replication. Make sense, right? So this is where G0 also can occur, right? But in G1, right, basically during this time, the cell, is growing, and so this is the cell growth part, right? So the cell is growing, and just like building up its resources, getting slightly bigger in size and getting ready to to go into cell division, right? So, for example, so during this time, basically, the cell is kind of doing what it's normally supposed to be doing, right? So, for example, liver cells are storing sugar, leaf cells are performing photosynthesis, all these kinds of things, right? So they're just going to keep growing during this phase until they get too large to continue normal daily activity, right? So we notice here. Remember we talked about there are checkpoints in the cell cycle, right? So let's suppose the cell starts growing and growing, and it gets like pretty big. It gets too big to continue growing. So first of all, why does it grow? Right? Anyone know like why does it grow? Freeha? Why is it important for the cell to grow before it divides? Mm-hmm. Yep. Mm-hmm. Okay. How about Yusuf? What do you think? Why is it important for the cell to grow before
Because
dividing?
as it splits, it's, I mean, like if it's, because when it's gonna like reproduce, it's just gonna split into two cells.
Mm-hmm.
It's
Right.
gonna get smaller.
Right. So when a cell divides into two cells, right? If it hasn't grown first, well then the two daughter cells are gonna be like tiny. They're not gonna have anything in them, right? So
Yeah.
the new daughter cells, they need to have all the complete, package. They need to have all the DNA,
Brother,
all the enough
what exact
enough mitochondria, lysosomes, everything that they need. They need enough. So
what
the initial parent cell, it's got to grow and produce all those things, so that when it divides into two, the new two daughter cells, they're big enough themselves. Otherwise, those are just be like tiny cells, and they're gonna have a difficult time growing. Yeah, you should go ahead.
brother, what exactly happens in the first checkpoint, the G1?
All
Like,
right, correct. So we look down here on this bottom picture, right? So in this checkpoint, all right. So what's happening? It's checking the cells, checking for to make sure that it's big enough, it has enough nutrients.
so this is all checkpoints do? G1, like for example, that's all it does. Just checks for
Right,
if the cell is good to reproduce or not.
right. So it checks there, and if it's not good enough, then it either stays in G1 or goes into G0. Otherwise, if it's good enough, if it decides, hey, the cell is big enough, we have enough nutrients, we have growth factors, the DNA is not damaged, then it goes into S phase. Right?
Hmm.
So in G1, this is basically like growing. It's growing and just forming larger inside. It's doing its normal work, but during the time, it's also growing and growing and getting bigger, so that eventually, you know, the daughter cells are going to have enough material themselves when they form. Right? So you have parent cell and then daughter cells. That's what they're called. Parent cells initial, and then the two daughter cells. Those are the results. Right.
Hmm.
All right. So here we have S phase. So S stands for synthesis. All right. So that's one way to remember, right? So S phase stands for synthesis. So in the S phase, right, what are they synthesizing, right? They're synthesizing DNA. They're synthesizing DNA, right? That's basically making a lot of DNA, right? So the chromosome. So in each cell, there's a whole bunch of DNA in the nucleus, right? So, but the cell, what the cell needs to do is it needs to copy the DNA and form double copies, right?
Uh,
So Anna, so why do you think we need to form two copies of everything during the S phase? Like, why is DNA replication important? Like, why does the DNA strands need to like kind of double, make copies of each other? Anas, what do you think?
Hmm. Probably they need to make like another strand so they. Mm, honestly don't know.
Not sure, no problem. Yusuf, what do you think?
because basically like the DNA it's gonna be used to make more cells
All right, Freya, what do you think?
like
Like, why is it
instruction.
that why is this that DNA replication is important here? Why do we need to double and copy the DNA in a cell? there's we're gonna form two daughter cells, right? So we need double the DNA, right? So each of the daughter cells is gonna have to have its own full copy, right? So right now we have one full copy, so now we need two to put one into each of the daughter cells. Make sense? Right? So now remember in S phase, right? In S phase, there's not like a checkpoint. You notice in this pink diagram at the bottom, right? There's not usually like any kind of checkpoint here in the S phase. We're gonna have another checkpoint when we get to the G2 phase, right? But here, the DNA is replicated, and we're gonna form double copies of the chromatin. The chromatin is like the term for like the DNA, like when it's kind of arranged in like a certain organized fashion. So that's basically kind of like another term for like DNA, right? So now we're going to go on to the G2 phase, right? So the G2 phase,
Brother, hold on, brother, hold on.
yeah,
So in the S phase, all that happens is the DNA is replicated, right?
right, right. Now in the G2 phase, right after the S phase, we go to G2 phase. So in the G2 phase, it grows a little bit more, all right, and it just kind of makes like a lot of different random preparations for for the cell to get ready to divide, because the cell is about to divide right at the end of G2 phase. So that's what's important now, right? So the G2 phase, right? This here at the bottom like gives a little bit of comparison of what's going on here, right? So in the G1 phase, there's kind of like a lot of general synthesis of proteins, RNA, stuff like that. Cell growth is occurring. In the G2 phase, the cell is already a big size. It's just kind of doing some final preparations, right? So it's more so it's not just growing in general now. It's growing and preparing certain things that are important for cell division specifically, right? In G1, there's kind of like general cell growth going on. Just generally the cell is kind of getting bigger, making a lot of everything. In G2 phase, it's growing a little bit, but mostly what it's making are the things that are specific to the specific cell division process that's about to occur. Right? So this is more specific cell growth.
Wait, but I thought in G2 and G1 nothing forms. It says over here that synthesis of proteins for spindle formation and mitosis occurs.
Right.
So in G2, like it doesn't check for anything. It just forms, like it just
No,
makes something.
there's a checkpoint, right? So remember there's one in G1, and then G2. Again, you see in this picture over here on the right, there's also a checkpoint there as well.
Oh.
Right? So there again, it's checking like are there any mistakes in the DNA? Is the cell big enough? Things like that. So in G2, there's also a checkpoint there. But you see, so it says synthesis of proteins for spindle formation and mitosis occurs, right? So these specific proteins are being made, right? The cell's growing, like these specific parts that are important for mitosis. Right? Any questions on that? So the cell is growing, but it's more like specific growth. It's not just
Like
it's not just generally
specific
cells.
areas.
So then what happens in G1? Synthesis of RNA and What's
and protein needed for cell growth. Protein.
All right.
Right. So in G1, the cell is just growing generally. It's not only making. It's not in G1, right? So take a look. Right after G2, you see like in this faded section here, there's mitosis and cytokinesis about to occur, right?
Yeah.
Right after G2. So in G1 over here in the beginning, right? It's just like general growth happening. Now in G2, now we're making things that are specific to the mitosis, like getting things ready for mitosis specifically. Right? So that's the kind of growth that's happening. It's like like what things do we need for mitosis specifically? Not just the cells just getting bigger overall, but like what are the specific things that we need to make for mitosis?
Okay.
You understand the difference?
Yeah, I understand the difference now.
Okay.
DNA mistakes, cell volume, and environment.
Right. Yeah. So there's another checkpoint in G2, and it's just making sure that we're ready to go to mitosis. So if that checkpoint passes, then the cell goes into mitosis. Right. So now we have mitosis and cytokinesis. Right. So this is where the cell specifically is going to be, tearing apart into two different things. So we're going to talk about, overall in this picture on the bottom, you see like some specifics right over here, right? So we're going to talk about this, in more detail in the next slides, right? But in mitosis, maybe you guys have seen this before: prophase, metaphase, anaphase, telophase. Maybe you
Yeah, I
guys have
did this.
seen this before in mitosis, right? Now, so this first part is mitosis, and then this last step, right? Now, over here, the cell is still connected together, right? Over here, in mitosis. In cytokinesis, this is where the cell is actually gonna divide into two. That process is called cytokinesis, where that last phase, where specifically the cells are going to divide, and then in the end you're going to get two daughter cells. Right. So let's talk a little bit more in detail about some of these things. Right. So first of all, we have
Brother,
mitosis.
in the S phase, when the DNA is replicated, right?
Mhm.
In the chromatin, the DNA is replicated. So, um, what's it called? Where is that DNA used? Like they still haven't talked about that, have they?
Yeah.
Yes. No. So in mitosis, you'll see like where it's used.
Oh, okay.
Yeah. So in mitosis, it's going to be divided up. So we're going to talk about that. All right. So first, let's talk about mitosis. Right. So we're going to go through the different steps of mitosis and cytokinesis. All right. All right. This is just like kind of like a review slide of the different um, phases of the cell cycle. All right. So let's just make sure we know specifically. So in G0, right? Remember, G0 is like the resting phase, right? That's where like nothing's really going on, right? G1, the cell increases in size, it grows, and there's a G1 checkpoint, right? In S phase, DNA replication occurs, right? DNA is doubled. And in G2, the cell continues to grow, and everything's just kind of like being made ready for mitosis, and there's a G2 checkpoint, right? So this is just kind of the same thing, it's just everything's on a table. All right. So they mentioned some like random facts here. I don't know if these are really important, but they're mentioning some random facts to give some perspective in terms of time. So it says not all cells have a life cycle of the same length. So some cells complete the steps of interphase in 8 hours, and others take years. So it says the cells have an average life cycle of about 16 to 24 hours. Right. So some of these cells are going through the whole cycle in about 1 day, and others are taking about years. Right. So the bone marrow goes through cellular division in 10 hours. This whole cycle it goes through in 10 hours, because that's always making more blood cells, white blood cells, all these kinds of things are constantly being made in the bone marrow. So for blood, the things that make blood, that's very quick. Now it says here your brain cells divide until you are about 7 years old, and then they slow down. Right? So other cells they're going to take like a lot longer to divide and make two cells. Bacteria go through the cell cycle every 20 minutes, while a fly embryo divides in 8 minutes. Right? So obviously, like bacteria, they're going to be like much more quickly, right? Because bacteria, for one thing, bacteria are much more simple, right? So they can go through much more quickly compared to like cells in the human body, which are more complex, right? So I guess this is just some random examples. I don't know if they're going to ask you about this, but these are just like some random examples, like certain things that are simpler, like fly embryos, bacteria, they're going to be go much more quickly. Bone marrow cells, where you need to create blood cells much more quickly, 10 hours. Brain cells are going to take a lot longer, right? So brain cells, they don't just keep replicating quickly, so they're going to go through this whole cell cycle, through this circular cycle. It's going to take them like a lot longer to go through. Right?
Brother, can you go back to the G2 phase image real quick? I just want to see something. Oh, that's way back. Oh, all right. So it checks. So it proteins looking for spindle formation. Okay, I see. So it synthesizes proteins for spindle formation, right?
So we're going to talk about what spindles are. We'll see what those are. So
Yeah.
spindles are important for mitosis. So we'll talk about what those are.
Oh.
Okay.
Okay.
All right. So let's take a look. So first of all, let's look at what is DNA and or like just like the structure of DNA and chromosomes. So in a in a cell, right? First of all, remember we have prokaryotic cells and eukaryotic cells, right? So, so Anas, which ones are human cells? Human cells are which one? Prokaryotic cells or eukaryotic cells?
Hmm. Proc. What are the options?
Prokaryotic and eukaryotic.
Probably prokaryotic.
Prokaryotic, all right. Yusuf, what do you think?
Brother, what's the question? Sorry, I was
So,
writing something.
so human cells are which type? Prokaryotic or eukaryotic?
Yeah, obviously eukaryotic.
So eukaryotic, right? So the prokaryotic cells are like mostly like bacteria and like so there's similar things to that. So prokaryotic cells are very small and very simple, and then eukaryotic cells are like human cells, mammals, plants, all these other kinds of things, are eukaryotic cells. Right?
Okay.
So in prokaryotic cells in bacteria, their DNA is arranged as a single circular molecule, right? So all their DNA is basically kind of like just like one big circle, right? Now it's kind of wrapped up a little bit, but overall it's kind of like one big circle, right? Now in a eukaryotic cell, right, it's much more complex. So this picture on the right kind of shows you how DNA is organized, right? So we see over here, we start over here, and first we see like the individual DNA bases and like the molecule, the double helix as we've seen it before, right? So if we kind of zoom out, you kind of see that it then kind of, you know, what happens is that the DNA is wrapped around proteins. So these proteins over here, in this like dark purplish kind of thing, this is these proteins are called histones, and they form something called a nucleosome, which you see over here on the left. So you can see over here when you zoom in, right, it has these histones. You see it says 8 histone core. So these are basically like certain kinds of proteins, and you see the DNA strands are like kind of wrapped around them, right? So they're kind of
Yeah.
wrapped around, and then each of these nucleosomes is then kind of wrapped around into another kind of helical kind of shape. And then eventually those helix are kind of like wrapped around themselves, and then those helices are then further wrapped around themselves. You see how so you see how like the DNA is like packaged. This is how the DNA is packaged to eventually form this chromosome. So this you see this picture on the bottom right. This is kind of
Yeah.
what you guys have probably seen pictures like this, right? So if you kind of like zoom out, or I mean if you zoom in one part at a time, this is how it's organized. So if you guys kind of see, right? At the beginning, you have the DNA. It's wrapped around these histones to form nucleosomes, right? And then the nucleosomes are then kind of wrapped around themselves to form these fibers. And then each of these fibers is then you see right over here is kind of wrapped around again, right? And then each of these things is then further wrapped around over here. And then eventually those are kind of sort of wrapped around over here. That's right, what happened? Right, this is the DNA, right? So you see on the top right, these colors. This is you've seen these. Have you seen like DNA pictures like this before? Right. So this is kind of showing you how DNA. So DNA is not just floating around in the cell. It's all tightly wound around proteins, and everything is wrapped up into an organized fashion. This is kind of what this is telling you. All right. This one over here, step 5. Right. These are DNA, but you see how like it's all being. You see how if you look closely over here, do you see how each strand is then like kind of being wrapped around to form kind of like a larger strand? Right. And then when you go over here to step 7, near step 7, you see how these strands are then wrapped around to form like bigger strands. Right. So it's like a series of like things being wrapped together. Right. Right. So this is just
So
kind.
so So a nucleosome is wrapped with histone, 8 histone proteins, right?
Right. So nucleosome contains these. So the nucleosome is the whole thing. So it contains all these histone proteins.
So what's wrapped around these?
Yeah. So you see this strand. That's the DNA. That you see this
Oh,
red
that's
blue. Yeah, that's
yeah.
the DNA being wrapped around to
Then what
hold
do
it.
the nucleosomes do? I've never seen nucleosomes in a DNA before.
Yeah. No. No. So the nucleosome is this whole structure. This whole structure, proteins and DNA put together, being held together. That thing is called.
Oh, a
Uh,
nucleus.
yeah. That whole thing is called a nucleosome.
So what do they do?
This is just a way of like organizing it so that the DNA is not floating around randomly.
Oh, all right.
Right. All right. And this is kind of saying the same kind of thing. I think this picture was not as good. That's why brought that other picture. But this is kind of saying the same kind of thing. This is just mentioning that overall we have 46 chromosomes, right? So if we go over here, I think on the next page, yeah. So over here on the next page, this is like these are all the chromosomes in the human body, right? So you have 1 through 22, and then the XY at the end. So you have 23 sets, and there's 2 within each, right? So that's 23 times 2, 46. Right?
Mhm.
So one set comes from the father, and one set comes from the. Hmm? So these are the chromosomes. So this is like the DNA all wrapped together, right? Do you remember at the bottom right over here? So this right over here is one set of chromosomes right here. So this right over here is all the chromosomes that you have. So each of these, like each of these things, like number 15, one of these things was like one of those wrapped up pieces of DNA that we saw,
Oh,
right? So all of this is like a whole bunch of DNA
so
that
brother,
is
the 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 chromosome from the father is highlighted in green, and the chromosome from the mother is highlighted in purple, right?
right, yeah.
So the last chromosome, the XY represents female and XX represents male, or is it the other?
Right. So XY means male, and if there are
Uh-huh.
two X's, XX, then that means female,
Uh,
right?
that's why only the father can they say that like create a male, right? Like he's in charge, like
Right,
he has
because
the
there's only
ability.
one Y chromosome there.
Yeah.
Hmm. Okay. So usually in the cell, so you notice over here, usually in the cell, it's not organized this cleanly, right? So these
Mhm.
pictures they were taken specifically in specific times during mitosis when everything is wrapped up in a super organized way. So usually things are organized, but it's not usually this organized, right?
Uh-huh.
So everything is get like super compact and organized when the cells are dividing. That's when they stain these and take a look at them. Apart from that, it's a little bit more difficult to see them. So they're still organized. The nucleosomes and everything are there, but they're not as wrapped up and compact as this most of the time, right? So now let's take a look at mitosis, right? So in mitosis, there's a whole bunch of different phases, right? So this is showing you some of the phases, right? So after interphase, you notice over here at interphase over here, this was like what we were talking about, G1, S, G2, right? So from there, if it's ready, right? If the cell is ready, it goes into mitosis. Remember at the G2 checkpoint. If it's ready, it goes into mitosis. So then in mitosis, you have prophase, right? Then you have metaphase, anaphase, telophase, and then you have cytokinesis, where the cell actually divides into two pieces, right?
Oh.
So let's look at each of
Uh,
these more specifically.
brother, hold up. Can I ask one question real quick?
Yeah.
Um, what's it called? So chromosomes are consisted of nucleosomes, right?
Right. They have nucleosomes within them. Yep. They're all wrapped up.
Like there's no count. They're just like made of nucleosomes, and
Yeah, you could.
nucleosomes are made of DNA,
Right.
and yeah. So then chromosomes are made of nucleosomes are made of DNA. I bet.
Yeah, you can see that. Right. All right, so let's take a look at each of these things a little bit more closely, right? So we'll go to specifically, and then we're going to go through again. And so at the top, this is like the cartoon pictures at the top, and at the bottom are like some of the stain pictures from like real stains from real cells, right? So in prophase, right? So what's happening in prophase? Basically, in prophase, this is when you start to see the chromosomes condense and become visible. So you remember those nice organized chromosomes where you can see each individual chromosome, right? So something like that happens in prophase where the DNA is really becoming compact and you can really see the chromosomes nicely, right? Now, you have these things, you notice these orange and blue things at the top left?
Yeah.
Orange and blue things, right? The orange things are called centrosomes. And then you have these spindle fibers coming out of them, right? So there's these like fibers that are coming out of them, right? And the nuclear membrane, it starts to break down. So this cell is starting to get ready to start to divide. So this is prophase, right? So you see the blue nuclear membrane that holds everything in the nucleus together, that
Yeah.
starts to break down so that the chromosomes can come out.
Brother,
Go,
how many chromosomes does one cell have?
so it's 23 sets, so total of 46.
So for humans, it's for 46 in each cell.
Right, in each cell.
Each cell, all right.
Right.
So when it splits, then each daughter cell has 23 cells, then, right? 23
Right.
chromosomes, right?
Right.
So then how do they get 46 chromosomes again?
Oh no no okay no okay so you have they have 46, but what it is that each of the 46 was
Uh,
doubled.
oh
Oh.
yeah, back
Remember?
during DNA replication, right?
Right. So everything was doubled.
In the S phase.
Right.
Uh,
So everything was yeah. So each of the 46 was doubled. So each daughter cell gets one of those. Right. Now, so this is prophase, right? So in the prophase, you notice the important part is that the DNA is still within the nucleus. It's still within that circle. Right. Now, what is the purpose of that spindle fiber? The spindle fibers. You notice there's two centrosomes here, right? So one is going to go to the left, and one is going to go to the right. So you notice over here, one is at the left, and one is at the right. So basically, what's happening is the spindle fibers is imagine like they're like ropes. Imagine they're like ropes, and they're being thrown at the chromosomes, and each one is gonna pull the chromosomes to one side or the other, right? So the spindle fibers, you see these blue fibers coming out. Those are
Yeah.
gonna like be those are kind of like ropes that go in and attach to all of these chromosomes right over here. Right, they attach to all these chromosomes.
So the blue the purple, sorry, so the blue colored things are the chromosomes, right? And then the green are the fibers.
No, no. So see these red things in the middle. These red things lined up in the middle.
Yeah,
Those are
yeah.
the
The
those are the
axis.
chromosomes. Yeah, the things that look like X's.
So then what are those green things? Those are like the strands, right? The
No,
spindle fibers.
these green things. No, so the blue. You see these blue lines?
The blues are the what they call the spindle
Oh,
Wait, which one are you looking at? This one down
fibers.
here?
yeah, I see it.
Okay,
Spindle
okay,
fibers.
yeah. So the same thing down here. This is the blue is the chromosome, and the green are the spindle fibers.
Oh,
Okay,
oh,
so
yeah. I was looking at this picture,
okay,
not at the
okay, yeah,
top
yeah.
one.
Okay, yeah. So the idea is that to each remember we doubled each chromosome, right? We replicate each chromosome, and
Mhm.
so you notice that the spindle fibers are going to come and attach to each chromosome from both sides, right? So Freya, what do you think happens? Like what is the purpose of that? Why is the centrosome from each side sending these spindle fibers to attach to the same chromosomes? What, so what do you think is the purpose of this? You see the spindle fibers are coming in and attaching to the chromosome from each side. So why do you think that's happening? They're trying to separate, right? Remember we doubled it, right? So when you attach from each side is going to pull one of those two chromosomes, and so basically it's going to be split in half, right? hmm Uh, yeah. Which one are you talking about? Are you talking about the ones up here or down here? So the. Mhm. Right. So, yeah. The red in the middle is the DNA, and the so no. So the chromosomes are made of DNA, right? So the chromosomes are made of DNA. So this red in the middle, these are chromosomes. They're made of the DNA. They're the same thing. So these blue are spindle fibers. Those are fibers that start to build up, and they travel and reach and attach to the chromosomes. Okay. Right. No, those are not DNA. So the blue, right? Only the red up here is DNA, right? So you notice from so this right here, you see that in metaphase they're all lined up in the middle, right? See in metaphase they're all lined up in the middle. So that's called metaphase plate, right? So the metaphase plate, they're all everything is like lined up in the middle. And you notice here in the picture down below, right? That's what it's showing. All the chromosomes are lined up in the middle, right? Now next, those um, those chromosomes are split apart, right? And so one copy is dragged to one end, and one copy is dragged to the other end, right? Right. So you can kind of see that this is so one cell is going to start to form over here in the right, and one in the left, and so that's what you see in telophase. In telophase, you see that just like in prophase, the nuclear membrane starts to break down. In telophase, is going to start to reform. You see the circle is starting to reform the nucleus on each side. So one into anaphase, when the chromosomes are broken apart and they're traveling towards one end or the other, once they reach each end, they start to reform their nucleus. All right.
Brother, so where do the centrosomes come from? Like, what are they?
So usually they're just hanging out in the cell until
Uh,
the cell
They're
cycle.
just vibing.
Right. So I
you-
don't think that they have any other function besides this. So they're just I don't think they do anything else usually. Right.
yeah.
So now in telophase, once the nucleus starts to reform, then you go to cytokinesis and you actually see this physical, separation between the cells start to occur. And there's a difference between animal cells and plant cells, which we're going to look at in a little bit. Right. So let's look a little bit more closely. This is the same thing. I think we went over most everything, but let's kind of look at it again. So in prophase, right? Remember in prophase, right? You're going to start to see the chromosomes inside the nucleus, and the nuclear membrane, the blue dotted line, is going to start to break down, right? And you see over here, these are the centrosomes with the spindle fibers coming out. So those spindle fibers are going to start to form. They're going to start to form. So over here, it says here that the centrioles are organelles which are only active during cell division. So this is the only time when they actually do anything, right? And so again, they produce these spindle fibers that go out and grab onto the chromosomes. And this picture down here just kind of shows you down here. This is kind of like from the electron microscope. This is the kind of picture, right? So
Oh.
these are the centrosomes or the centrioles right over here.
So centriomes, centrioles.
Yeah, centrioles. Hmm. Right. So so they could. So like for example, in this so they probably not. So here's what they do, right? So they um, so these are probably different cells, right? Like if you look at the pictures at the bottom here, these are probably different cells, and so what they probably see is they see all the so if they have like enough cells, right? You're gonna see some of the cells going through all of these different phases, right? I'm not sure. I'm not sure if they. I don't know if they see like a cell that's alive going through this because you might have to stain it. I don't know if there's some stain that they can put in where, as the cell is alive, you can see like what's happening inside the cell. I think it's possible. I'm not sure. So these cells are probably dead because they've stained them. See all these like fluorescent stains. So I don't think
Yeah.
these cells are alive down here.
But
So,
but
so I
like
don't-
this is colored stains, right? This isn't metal stains. So I thought in
Right.
like only in metal stains they die, right? Not in color stains.
right. Yeah. So in metal stains they die, but in this is also this is fluorescence. So the cells
Oh.
also die in this one as well.
Okay.
Freya, your
So he
question?
actually saw all of this. That's just crazy, really.
Sorry, Freya, you had a question? No. Okay. All right. So now this one over here, this picture over here on the bottom right, this is from electron microscope. So the cells are dead here also.
Hmm.
Right.
Brother, can you go back to that picture with those pictures of this mitosis occurring? Oh, okay. So that's like showing the that's showing you the like the what's it called? Zoom how much they've zoomed in, right? The 5 and then they forgot what that U stands for.
Right. That's going to show you how long it is. I mean, yeah, the size.
Oh.
5 p.m.
So let's take a look at metaphase, right? So we looked at metaphase. So in metaphase, we form this metaphase plate. You notice all of the cells are lined up here in the middle. That's called metaphase plate, right? And so over here, down here, you see in the middle, right? All of these are lined up, and you see the spindle fibers coming out over here.
Yeah.
Then next in anaphase, during anaphase, they've been pulled apart, and the chromosomes are kind of traveling towards one end or the other end, right? So you see now they're in two groups. So these ones over here are going to eventually form like their own cell, and these ones over here are going to eventually form their
They're
own
probably
cell.
even dead because they stained these ones.
Right, yeah, so I think these cells are probably dead because they stained them, right? So this is called anaphase. So it says the centromeres that are holding the sister chromatids together divide, and the individual chromosomes are pulled apart, right? So the chromosomes, each copy is like pulled apart to from one end to the other, right, by the spindle fibers. So in telophase, right, in telophase, basically the nucleus starts to reform. So you see this like circular kind of thing that's starting to reform on each end. So that's where the nucleus is starting to reform. The nuclear membrane starts to reform, and everything is starting to become more compact and organized again. Right? Now in telophase, we haven't split up yet. We haven't split yet, right? So that part is called cytokinesis, right? So cytokinesis is specifically when the cells actually, split into two, right? So this is called cytokinesis.
Wait, so the telophase is the last step, and then after that, cytokinesis occurs when they split, right?
Right. So
Yes.
cytokinesis is not usually considered part of mitosis. Right. So there's a difference between cytokinesis in animal cells and plant cells, right? So in animal cells, right, the different proteins act in myosin kind of pinch it off, kind of pinch off the two cells into two separate cells. Okay.
So they like just cut it.
Right. Because in animal cells, what is present in plant cells that's not present in animal cells?
Cell wall, brother, cell wall.
Cell walls.
Cell wall, right? So do you think, Freya, in plant cells that we can do the same thing? We can just pinch it off and form two cells?
No, they can't do that because the cell wall is gonna.
No, right? Because the cell wall is very strong, right?
Oh.
So you can't just do that to the cell wall. So in plant cells, right? Basically, a nucleus forms on each side of the old cell, and then it doesn't pinch off. Just in the middle, it starts to just build like another cell membrane and cell wall in the middle. Right.
Oh.
So that's called a cell plate initially, right? So it doesn't pinch anything off, and they move off separately. It just everything moves to one side or the other side, and it starts to build like kind of like another cell wall in the middle. All right. So the reason is because in plant cells they have the cell wall that's not as flexible as just like the cell membrane in animal cells. All right. All right. So now, so in cancer, right? so this is just like last like two slides. So this is what happens in cancer, right? So cells are dividing, right? See over here on the left, cells are dividing, right? But sometimes in the DNA, right? The DNA is always checked to make sure that it is correct. But sometimes there are problems in the DNA. Sometimes there's a mutation that occurs, right? I know. So what is a mutation? Have you heard the term mutation before?
It's like something like turning into something else.
Mm-hmm. Right. So something turning into something else, right? So mutate means like something changes. So the
Yeah.
DNA should be like copied exactly correctly, right? But sometimes there are changes that occur, like there's a mistake that's made when you're copying the DNA. Remember in the S phase when we're copying the DNA? So sometimes there's a mistake that's made, like the wrong base is put in. And so what this is showing on the left is that sometimes when you have a couple of mutations, you form a cancer cell because this cell over here doesn't work properly. It doesn't,
Mm.
the DNA usually gives you the instructions, hey, if there's a problem, then you got to stop. You can't divide. But in a cancer cell, maybe there's a mutation that tells it to ignore the problem, and so it just keeps dividing anyways, even though there's
But
a problem.
but so for every person who has cancer, then their issue is slightly different from someone else because
Exactly
the
right.
cells act in different ways, right?
Right. So that's why there's not like one cure for cancer kind of thing.
Oh,
Because
okay. That makes it so more
because
complicated.
every mutation could be different, right? There's like always a different
Yeah,
cause.
exactly. Yeah.
Even within like liver cells or skin cells, like many different kinds of cancers, right? So and they're all caused by some different mutation, right? They could be mutations all over the place in random things. So that's why there's not like one cure for cancer. That's why everything is like different, and so it's like so there are certain cancers where like they found like like one medicine like 98% of the time will cure it, but then other cancers like maybe like only 10-15% might live.
Oh,
So
all
that's why
right.
it's always different. So here's what happens in a normal cell. Let's suppose there's some problem in a normal cell. The cell basically kind of kills itself. This is called apoptosis. So the cell recognizes, hey, there's some big problem here, and this cell just automatically commits this thing called apoptosis. It kills itself.
Does it commit suicide?
Mm-hmm. That's called cell suicide. So, that's right. So in a
Yeah.
cancer cell, right, even if there's some big problem, maybe the DNA mutation that was supposed to, give it the instructions, hey, commit apoptosis, it ignores it and it just keeps dividing and just keep going on. So that could be a big problem, right? So here, right, that's what it's showing at the top. Controlled cell death. That's called apoptosis. So there's a very controlled kind of cell death to get rid of this cell that has a problem. But in a cancer cell, maybe that doesn't happen. They say, oh, there's a problem, but it just ignores it and it keeps just dividing and dividing, growing and growing, and just taking over, entire organs, maybe spreading to other parts of the body through the blood and stuff like that. So if you have a cancer of the liver, it could, invade the blood vessel. Maybe this is growing so much that it goes into the blood vessel, and then from there the cells, can float off somewhere, and maybe they lodge somewhere else, and then you can have cancer that same cancer shows up somewhere else in the body.
So too much cell growth can also cause tumors, right?
Right.
That's not cell mutation, right? Because that's
Yeah.
So usually those are the result of cell mutations.
Yeah.
yeah, but then wouldn't that also cause cancer?
Yeah, yeah. So
Because
so so
cancer
there
is cell mutation.
Cancers, the like
so you
one cancer cells keep reproduction.
right. So
Yeah,
within,
but
yeah. So that's
yeah,
so that so there's so within so it depends. So there's different kinds of tumors and different kinds of cancers. So if you can have something called a benign tumor. That means that the cells are growing, but basically they're not doing anything to anything except for they're just taking up space. Right. So okay, you have a lump there or whatever, it's taking up space, but it's just mostly like an annoyance.
uh-huh.
However, you can have like a malignant tumor where it's growing, but now it's invading into places where it's not supposed to go. It's not just kind of like pushing against the outside. Now it's invading and taking over and damaging places.
Yeah, it might stop blood flow too, right?
Yeah, it could do that, something like
So
that.
what's the difference between cancer and that then? Because cancer does the same thing.
Yeah. So in cancer they call it cancer because if it's if it's not just kind of growing in one place, but if it's damaging the environment around it.
Oh, all right.
Also, there's also a difference between a cancer that might grow locally, maybe it's just invading locally, but it's not floating through the blood to somewhere else,
Oh,
right? So like even within cancers and stuff, there's like but the thing is so let's suppose you have a benign tumor, right? Let's suppose you have just like a whole bunch of cells that are growing, but
Mhm.
it's not really invading. It's not like damaging something. Okay, even though that itself is not considered cancer, that's like the first step towards cancer. You understand? So so even though you could have a tumor that's not cancer, but if that's the first step, then it just makes it easier for it to get to cancer.
oh,
Yeah.
You understand what I'm saying, right? Like maybe there's a mutation that stops it that that was supposed to stop it from growing, but the mutation is now causing it to grow, even though it's not really causing damage to the outside. It just kind of just kind of forming a lump and just pushing against the outside, not invading. But that's like the first step, right? That's the first step to get to cancer. So that even so, that's why if you have a benign tumor, they still recommend you take it out because you have a higher risk of getting into cancer because the first
oh
step
yeah.
has already been overcome.
For benign tumors, they like, but even if they like, I don't know, like cut it off or something, I don't know.
Hmm.
It won't stop the cell mutation, right? I mean, or the cell. Even though it's a benign tumor, it won't stop the like cell replication that's just going off.
No. So it will if you take out the whole tumor. So if you leave part of it, then it could regrow back, right? That's why
Oh,
you
so
got.
you take out the source, is what
Yeah,
you're saying.
exactly. You have to take out the whole thing,
Ah.
right? So yeah. So sometimes during surgery, if they leave part of it behind, then it could regrow back.
Oh.
Right? So even in a cancer, let's suppose you have a cancer, right? If you don't take out every cell, then it's going to regrow back, right? But so that's
Oh.
why
Yeah.
during surgery though, right? You can't. I mean, it's going to be very difficult to take out every cell, right? You can't like microscopically see every cell in a surgery.
Exactly.
So that's why they recommend like surgery to take care of most of it. But then all the microscopic cells that you can't see that are left, you got to still give medicine to try to kill those off. So you have to use combinations.
Oh. So it's like forced apoptosis, but forced apoptosis.
Yeah, you can say something like that. Yeah. All right. All right. So I guess we'll stop there for today. So I think it's already late, right? So we'll have to try to do the quiz. Yeah, it's already 10:10. So we'll have to try to do the quiz next time. So I think this one also. There's like a lot more. There's like a lot of things to remember. So we'll have the quiz like first thing, inshallah, next time. I think on Tuesday we'll have the quiz for this one before we do the next lesson. All right.
Right.
So I'll send you guys this PowerPoint, and then the link to
Okay.
and then so the resource page, the student resource page. I'm gonna put the link to the recorded video on it.
Okay.
All right.
Anas, did you do the 2.7 assignment?
Yes.
Was it easy or what?
Wait, let me